Well, good morning. I'm sorry we're at a little rough start this morning, but we're delighted to have you join Asthma Grand Rounds. I'm Chris Fanta, substituting for Elliot Israel this morning. And we're delighted to have as our speaker Dr. Joshua Boyce, whom most of you already know. He is the Albert Sheffer Professor of Allergic Disease at Harvard Medical School. He heads the Jeff and Penny Vinnick Center for Allergic Research uh, at Brigham and Women's Hospital. And what you may not know is he is the 2024 Harvard Medical School Joseph Martin, the recipient of the Joseph Martin Award for Advancement of Women at Harvard Medical School. It's a terrific honor. And we're delighted to have him here to speak this morning. CME credit by sending your name and degree to Dr. Israel's assistant at eIsraelassist at bwh.harvard.edu. And Harvard is awarding CME credit at the end of the year, at the end of our program in June. And then you'll receive all of your credit. And so without further ado, thank you, Josh, for giving our asthma grand rounds today.
Well, thanks, Chris, for the nice introduction and for the invitation to talk today. And my topic area is mast cells in asthma. And this is a topic area that's undergone a number of changes in public opinion over several decades, dating back to a period where mast cells were broadly regarded as likely important in asthma, to a period of a couple of decades where they're essentially were ignored as the field focused on other features of type 2 inflammation. But in the last 10 years, we've accumulated a body of evidence that strongly links mast cells to asthma pathogenesis and makes them likely therapeutic targets. What I'm going to do today is tell you that this is a nuanced story, and the role of mast cells is likely to depend on a number of host-specific factors that have to be taken into account as we consider options for clinical trials in heterogeneous populations of patients with asthma, and patient selection will be absolutely essential. So my order of presentation, if I can get this to work, let's see. I'll start with some basics about mast cell biology and the history of mast cells, which is quite interesting. I'll talk about mast cell heterogeneity and how that concept relates to the populations and subpopulations of mast cells in the asthmatic airway. I'll discuss two intertwined stories relating to the role of mast cells in asthma, IL-33 and prostaglandin E, and we'll see how those are intimately tied. I'll talk about some lessons from clinical trials that have been completed, and we'll finish up with some conclusions. So mast cells were a discovery of Paul Ehrlich during his experiments as a medical student in the late 19th century, and he recognized mast cells as a uniquely staining cell that he found in every peripheral tissue in human pathologic specimens. He called them mast cells from the German word to masticate, because he interpreted the blue cytoplasmic inclusions as something the cell had eaten or consumed. But of course, these are secretory granules. Now, the fact that mast cells are exclusively resident to the tissue led many to suspect that they were mesenchymal derivatives rather than hematopoietic. And it actually took 100 years for Kitamura and colleagues to unequivocally prove that mast cells came from the bone marrow. The reason we never see them in the circulation is they only circulate as exceedingly rare committed progenitors. The mast cell progenitor accounts for maybe 1 out of 20,000 peripheral blood leukocytes in healthy individuals, has no distinguishing morphologic features, and can only be recognized by flow cytometry based on CD34, CD117, which is the KIT tyrosine kinase, the high-affinity IgE receptor, and the beta-7 integrin, which mast cells use to migrate into mucosal sites. But only when they hit the tissues do they become these fully granulated, very distinctive effector cells. Now, mast cell development has an absolute requirement for the membrane-bound growth factor, stem cell factor, and the KIT tyrosine kinase, CD117. And this has direct therapeutic implications, as I'll explain later. Now, mast cells existed long before Paul Ehrlich. They emerged probably during invertebrate development because they're found in tunicates, which are about 700 million years old. So they are truly one of the immune system's most ancient cellular constituents, evolving hundreds of millions of years before there was IgE or even adaptive immunity. And clearly, evolution does not retain something so strongly and for so long unless it has some sort of advantage to the host. But one of the perplexing things about mast cell biology is we still don't know what its physiologic functions are. We only think about it in terms of disease. Mast cells are also perhaps the most unique of all hematopoietic cells. And this is beautiful data from a paper that came out in Nature Immunology almost 10 years ago from the InGen Consortium with Dan Dwyer and Nora Barrett leading the way. And these investigators did the first RNA sequencing studies on tissue mast cells that they sorted out of a number of murine tissues. These were naive mice, and performed bulk RNA sequencing. And what you see in this heat map is that the mast cell transcriptome in the upper right does not resemble any other cell. There are thousands of genes that mast cells and only mast cells express, indicating, again, a highly specialized function. And in the lower left, this three-dimensional principal component analysis shows you that computationally, the mast cell's gene expression pattern sits out, way out, compared to all other hematopoietic cells. It's an outlier. So it's functionally exceedingly unique. But about all we know about mast cells is their role in type 1 hypersensitivity. And this is a process that's really a misdirected adaptive immune response involving IgE and relevant to all allergic diseases, including allergic asthma. And classically, when mast cells see an antigen to which they have a specific IgE bound, they expel granule components, including histamine and proteases, and they generate eicosanoids, arachidonic acid metabolites. Now, I've highlighted in red the classical bronchoconstrictors. Mast cells are probably the most primary source of classical endogenous bronchoconstrictors, and it's no surprise that when an inhalation challenge is performed, you get this drop in FEV1, which relates to these actions of these mast cell-derived mediators. And then mast cells go on to synthesize and secrete cytokines, chemokines, and growth factors that can promote the recruitment of blood-borne leukocytes, which of course is a characteristic of the late phase response. But when we think about asthma, it's much more complicated than IgE. And many patients with severe asthma don't make IgE to specific allergens. But mast cells can be instructed by a broad range of innate immune stimuli. They're charter members of the innate immune system, so they can recognize microbial constituents, complement fragments, neuropeptides. And very importantly, mast cells are also a primary site of action of interleukin 33 through the ST2 receptor. I'm going to come back to that story later on because I think it's a very important part of what mast cells are likely doing in asthma. So this is a beautiful piece of data from a classical paper that's almost 40 years old. And this figure still gives rise to board questions, at least in allergy, maybe in pulmonary too. And this was a study where Donald Cockcroft used the classical inhalation allergen challenge model in subjects with allergic asthma who were dual responders. That applies to about 50% of atopic asthmatics. And he showed in this beautiful paper that sodium chromoglycate, the mast cell stabilizing drug, if delivered prior to the allergen challenge, blocked both early and late phase changes in FEV1. Beclomethasone, on the other hand, only blocks the late phase, albuterol only the early phase. Classical, beautiful experiment. And this type of model was used as a piece of evidence supporting the role of mast cells in asthma and the role of cromolyn as a treatment for asthma. But it turned out this isn't a very good model. For one thing, you can make it happen even in somebody with allergic rhinitis who's never wheezed a day in their life. It's not an asthma model per se. It's a model of what happens when you IgE crosslink the mast cells in your airway. So it's only reflective of the IgE-driven effector pathways. And it shouldn't be a surprise. It's a lousy predictor of drug efficacy. Cromolyn doesn't work very well. That's why we don't even have it anymore in an inhaled form. And so with the recognition of these caveats, the field began to move away from the view that mast cells were a principal effector cell in asthma. Okay, so with that as a backdrop, let me now introduce the concept of mast cell heterogeneity. And I like to look at this in evolutionary terms because the innate immune system did not have many options to create functional diversification. There were no antigen receptors to mutate, no T cells. And so to create diversity, it had to have cells that were versatile. And those cells really are mast cells and macrophages that have the greatest degree of transcriptional versatility. If you look at mast cells and macrophages across different tissues, they're entirely different in terms of what they express and probably how they function. Now, the most obvious feature of mast cell heterogeneity in the human is differences in protease content. About 25% of the mast cell's total weight are proteases that are stored in the granules. All mast cells have tryptase, and some mast cells have chymase, carboxypeptidase A, and cathepsin G. And thus, the designations of MCT and MCTC mast cells emerged in the 1980s because of the work of Larry Schwartz, and these designations have stuck. So there are some interesting functional differences between these two subtypes of mast cells. MCT mast cells depend on T cells. So T cell immunodeficiency produces a depletion of MCT mast cells, but not MCTC mast cells in the airway and in the gut. MCT mast cells tend to live close to or within epithelial surfaces in mucosal organs. MCTC, on the other hand, tend to reside in the submucosa around blood vessels, within nerve fibers, and in some instances within mucus-producing glands, as shown in this nice picture from a Rob Schleimer paper several years ago. So they differ anatomically, they differ in T cell dependency. Another important point is that MCT mast cells expand consistently in the mucosal surfaces when there's type 2 inflammation, whether it's type 2 high asthma, nasal polyposis, allergic rhinitis, eosinophilic esophagitis. All of these are diseases where you see MCTs crawl into the epithelium and expand. We'll talk more about that in a minute. Now, conveniently, the mouse has some parallels to the human. Those of us who work in preclinical models are happy about that because it implies that these features are largely conserved. The proteases don't match up, but in the mouse, the mucosal mast cells essentially are functionally equivalent to MCT in that they are T cell dependent and expand in type 2 inflammation in the epithelium, and the MCTC are more like the connective tissue mast cells in the mouse. I'll point out that in the mouse, because you can do these definitive experiments that you could never do in humans, that we know these two mast cells come from different lineages, interestingly. The mucosal mast cells come from circulating progenitors and have a short half-life. They're turned over every few days to weeks, whereas the connective tissue mast cells derive from embryonic primordia and almost never are replaced during adult life, at least in the skin, which is the organ where it's best studied. Now, I'll tell you, having gone to all the trouble of explaining these differences, that these characterizations vastly underestimate the true extent of mast cell heterogeneity. And I'll show you a very specific example of that in a moment. And this is because mast cells are highly sensitive to their environment. They receive cues from the extracellular matrix, integrins, cytokines, lipid mediators that can change their function in accord with their location and the needs of the host at that time. Very versatile cell type. Okay, so what do these subtypes look like in the asthmatic airway? We'll start with the MCT story. And again, MCTs like the epithelium and they expand during type 2 inflammation. So you find intraepithelial mast cells in high frequencies in patients with type 2 high asthma, but you also find them in non-asthmatic eosinophilic bronchitis. So this is really a reflection of type 2 inflammation, not so much necessarily specific to asthma. They do correlate with the type 2 epithelial gene signature. So the genes that are driven by IL-4 and IL-13, the so-called type 2 high genes, correlate with the numbers of intraepithelial mast cells. Their numbers predict steroid responsiveness. So the more of these mast cells you have, the greater the delta FEV1 when you give somebody inhaled steroids. And they themselves involute with inhaled steroid administration. And their numbers in asthma predict exercise-induced bronchoconstriction and cold air hyperreactivity. And we know that thanks to the work of Teal Halston's group in Washington. Now, what about the MCTC? The MCTC story became very interesting a little over 20 years ago because of a classical New England Journal paper by Ian Pavord and Chris Breitling, who showed that in the airway smooth muscle bundles, you have these MCTC mast cells. And they are specific to asthma. You do not see those in eosinophilic bronchitis, and for the most part, you don't see them in healthy controls. They're present in both type 2 high and type 2 low groups. So this is really an asthma thing, not a type 2 thing. They're relatively steroid insensitive, although high doses will bump their numbers down a bit. And their numbers correlate with both disease severity and hyperresponsiveness to methacholine. So we have at least two subsets that are histochemically, anatomically distinct that correlate with different features of disease activity. So it's one thing to count numbers, but it's quite another thing to understand what they're actually doing. And we're pretty limited in our ability to do that in the lower airway. But we do have some surrogates that are reflective of mast cell activation, and perhaps the most robust and specific is tryptase, specifically activated tryptase or beta tryptase. And this is from a study by Larry Schwartz and some colleagues who used a novel monoclonal antibody directed against active tryptase to look at levels of active tryptase in BAL fluids from patients with asthma across a range of disease severities. And you can see that there's a very clear severity-related increment in BAL fluid tryptases, the highest levels being detected in severe asthma. And even in the plasma of patients with severe asthma, you can pick up a little bit of a bump in tryptase, so that there seems to be a systemic leak phenomenon. Now, importantly, in this study, the investigators found no differences in BAL fluid tryptase between those individuals with severe asthma who had high blood eosinophil counts and those who had low blood eosinophil counts. So again, this feature appears to be not a type 2 high or low phenomenon, but rather a feature that cuts across all types of severe asthma. But are all mast cells doing bad things in the airway? Maybe not. This is data from Sally Wenzel and her colleague in pathology, Sylvia Balzar, who published this paper about 20 years ago in the Blue Journal, where they did a very careful morphometric analysis of bronchial biopsies from patients who were enrolled in the SARP protocol. And they looked specifically at small airways, so a different anatomic part of the lung. And they found that the density of mast cells in the small airways was very high compared to other airway calibers. Most of these are MCT mast cells, you can see in these black bars, but a fair number were MCTC. And they found, to their surprise, that the numbers of MCTC mast cells in the distal small airways actually correlated positively with measures of airflow, percent predicted FEV1 and FEV1 to FVC ratios correlated directly with the numbers of MCTC mast cells in the small airways. Now, this doesn't mean cause and effect necessarily, but it does suggest that mast cells in different anatomic regions of the lung probably have different functions as it relates to the airway. So we need to understand a lot more about the heterogeneity of mast cell subpopulations. And it's very hard to do that in the lung when you're taking samples from living individuals, but you can do it in the sinonasal cavity because of the fact that patients who have bad sinusitis, especially nasal polyposis, will frequently undergo surgical excisions. And this is from a beautiful paper by Dan Dwyer that came out in Science Immunology 3 years ago. And this was really the first use of single-cell sequencing technologies to try to understand the extent of mast cell heterogeneity and function in a human tissue. And what we're looking at here, this blob here in the middle of the slide, is a UMAP plot, which shows you 4 different color-coded, transcriptionally distinct mast cell populations from human nasal polyps. So there's way more than MCT and MCTC here. We have populations that we haven't accounted for. There is an MCT pole to this continuum. It's the red cells up here, and they are enriched for the enzymes of the leukotriene synthetic pathway and interleukin-5. The MCTC pole down here differs from the MCT pole by about 300 genes, including IL-13 and chemokines that attract Th2 cells and monocyte macrophages. So the two populations that we can recognize by antibodies clearly have different functions in the polyp. Then you have a population that sits in the middle, which is intermediate. We call these transitional mast cells, which we think are seeding both of these poles, perhaps a differentiation state. And then we have this purple population, which stands out because of its expression of proliferation-associated transcripts. And this is a very important point. Mast cells are unlike any other granulocyte because they proliferate in the peripheral tissues. And this is probably part of the way that they expand in type 2 inflammation. And the number of proliferating mast cells correlates, in sinus disease at least, with disease severity. you see the groups broken up here in terms of AERD. These are the folks who have the worst nasal polyposis, and often they have bad asthma. They have the highest frequency of Ki67 positive mast cells, which is a flow cytometric indicator of proliferation, compared to aspirin-tolerant polypoid controls and non-polypoid samples. And when you look over here, you see the proliferating mast cells correlate with blood eosinophil counts. So it appears it is a feature that is linked to the strength of type 2 inflammation, and polyposis generally is a type 2 high disease. So mast cells, pretty complex in the polyp. Can you infer anything from these data to mast cell populations in other tissue sites? And the answer is perhaps not. So this is a comparison that Dr. Dwyer did between the transcriptomes of skin mast cells from a number of public data sets covering a number of diseases as well as normal healthy skin versus the MCT and MCTC pools from the nasal polyp. And the heat map shows you that the skin mast cells, which are classically pure MCTC, bear zero resemblance transcriptionally to the MCTCs in the polyp, certainly not to the MCTs in the polyp. The point is that functionally, the histochemistry does not tell you what the cell is doing. And MCTCs in one tissue may be a completely different cell type from MCTCs in the other tissue. One of the most graphic examples is receptors expressed by skin mast cells, including MRGPRX2, which is for neuropeptides, is completely lacking on the nasal polyp mast cells. So histochemistry does not tell you about function, and we have major gaps in our knowledge. Okay, so now I'm going to talk about one of the factors that influences mast cell function, and that is IL-33. And hopefully by now, everybody's heard of IL-33. It's one of a triad of cytokines produced by mucosal epithelial cells. IL-33 is a DNA binding protein stored mostly in epithelial basal cells in the airway and endothelial cells. And when these cells are damaged or activated, interleukin 33 is released by mechanisms that are still not 100% clear. They act on tissue-resident innate immune cells, mast cells, ILC2s, perhaps macrophages. And they also, IL-33 also stimulates the activation of recruited eosinophils and basophils, as well as populations of Th2 effector cells. So it's a very efficient way of activating all of these important hematopoietic cells that drive type 2 inflammation. And the result of this activation is a secondary wave of type 2 cytokines and chemokines that promote the features that we all recognize associated with type 2 inflammation, IgE class switching, eosinophilia, epithelial and stromal cell functional changes, and end organ dysfunction. And the importance of this pathway in animal models is very, very clear. But actually, unlike many type 2 cytokines, IL-33 and its receptor ST2 did not enter the map because of animal studies. It entered the map because of GWAS studies in humans, where IL-33 and the locus encoding ST2, IL-1RL1, were reproducible, highly reproducible GWAS risk hits in asthma and also in chronic rhinosinusitis with nasal polyps. There were some experiments of nature in human populations. There's a loss of function mutation in IL-33 that produces eosinophilopenia and protects Northern European populations from asthma risk. And recently, there has been at least modestly promising results from clinical trials involving anti-IL-33 and anti-ST2 monoclonal antibodies. And it appears that the efficacy signal in terms of changes in annualized exacerbation rates applies both to type 2 high and type 2 low patients. So there's reasons to be interested in this axis. And I'll tell you that most of the literature focuses on the role of IL-33 as a driver of ILC2s, but I will tell you the mast cell is probably just as important. Now, these are data from two beautiful studies, both RNA sequencing studies, one done on nasal polyps by Dan Dwyer and Jose Ordovas Montañez that was published in Nature a few years ago, and the other one from Sarah Teichmann's group in England looking at asthmatic bronchial biopsies. This was published in Nature Medicine. And both of these show you UMAP plots. These blobs constitute transcriptionally distinct cell subtypes. And in both of these tissues, the mast cell cluster is readily identified because they're very transcriptionally active. And the orange tells you where the strongest expression of IL-1 RL1 transcript is, the transcript encoding the IL-33 receptor ST2. And you see the mast cell really dominates. It's the principal site of the receptor expression. And on the right-hand side of this slide, you can see that the mast cells in the nasal polyp clusters that Dan Dwyer identified are highly enriched for transcripts that are hypothetically driven by IL-33, including a number of important cytokines and chemokines, as well as cyclooxygenase 2. So there's reason to believe the mast cell is prominent among the cells that are targeted by IL-33 in this inflammatory milieu. Now, additional evidence for the involvement of IL-33 as a driver of mast cells in asthma specifically comes from the UBioPred Consortium. This is from a Blue Journal paper a couple of years ago, where the investigators collected induced sputum from subjects with asthma, mild to moderate and severe, and performed RNA sequencing on the sputum cells, and then queried the data looking for genes that are characteristically upregulated by IgE receptor cross-linking of mast cells or by IL-33 stimulation of mast cells. So these are mast cell transcripts induced by these two stimuli. And on top, you can see that the Fc epsilon induced mast cell activation gene signature is detected in asthma, higher in severe, moderate in the mild to moderate. The IL-33 gene signature in the bottom is most strongly represented in those with severe disease. When you look at the subtypes of sputum granulocytes, so they've broken it down into pauci, granulocytic, eosinophilic, neutrophilic, and mixed, standing for eosinophils and neutrophils. You can see that there's the greatest enrichment for the IgE-driven activation signature in those with eosinophil-dominated sputum. The IL-33 signature actually is greatest in those with neutrophilic disease and mixed granulocytic, and these tend to be the sickest asthmatics. So IL-33 is almost certainly affecting mast cell function in asthma, especially in those who have bad disease. So what is controlling the IL-33 compartment? I'm now going to introduce our last player here, and that is prostaglandin E. Prostaglandin E is the body's most abundant arachidonic acid metabolite. Every cell can make some, and this has been used, repurposed by evolution hundreds of times. Prostaglandin E can be very pro-inflammatory, but in the airway, it has a privileged role. It's a bronchodilator and an anti-inflammatory icosanoid. It inhibits the activation of mast cells, eosinophils, basophils, ILC2s, and it inhibits 5-lipoxygenase activation. So it attenuates leukotriene production. So you could view PGE as sort of an endogenous check against excessive type 2 inflammation that contributes to airways dysfunction. And there's actually very good human data to indicate this is exactly what prostaglandin E does. This is from an old study by Gail Gavreau and Paul O'Byrne, a classical allergen challenge paper where they show that PGE inhalation blunts the early and late phase of the response, also attenuates the recruitment of eosinophils and basophils to the airway. Chris, you'll be amused to remember that Paul O'Byrne, when he came as asthma visiting professor, showed this slide. It was a brand new paper at the time. And so PGE definitely inhibits the inflammatory response downstream of IgE-dependent mast cell activation. Now, it turns out that patients with respiratory disease have a broad range of PGE production capacity. And this is best known in the upper airway, where again, you have access to sinonasal tissues that are surgically excised. This is data from a Japanese otorhinolaryngology group who directly measured lipid mediators in freshly excised sinonasal tissues. And what you can see here is they've divided the patients up into 4 groups, 2 of whom have asthma and polyps. And you can see that in AERD, there's tiny quantities of PGE in the tissue relative to head and neck surgical controls and chronic rhinosinusitis without polyps. And you can see it's also quite low in those who have asthma, but aspirin-tolerant polyps. And the inverse is the case for the cystinoid leukotrienes. So very low PGE, very high cystinoid leukotrienes, perhaps directly related to one another. We don't have as much good data from the lower airway, but we do have data from some induced sputum studies, and this is from a Spanish group who reported in Allergy a few years ago that PGE in the sputum of patients with eosinophilic bronchitis is quite elevated compared to healthy controls, but you see almost none of it in asthmatic sputum, whereas you see this sharp increase in leukotriene C4 in the asthmatic sputum compared to the healthy controls. So again, evidence that PGE production is low in these disease states in the airway. And what might be the consequences of that? So that's a question that my lab has focused on now for several years. And I'm going to show you some data from a recently published paper by Kenan Al-Halak, who was a postdoc in our lab. And Kenan was using a strain of mouse, the PGE synthase knockout. This is a mouse that lacks one of the 3 PGE synthase enzymes. It can make PGE, it just can't increase the PGE production with inflammation. And so it's a nice model to look at the impact of PGE deficiency on airway pathology. And in this house dust mite-induced model of pulmonary inflammation, you can see the PGE synthase has higher BAL fluid total cell counts in eosinophils, higher numbers of proliferating and activated group 2 innate lymphoid cells. Those are major sources of type 2 cytokines in this type of model, and higher levels of lung IL-33 protein. So you reduce PGE production capability, and all of these features of type 2 inflammation are amplified. Now, we were interested in what was happening with the mast cell compartment. And so in collaboration with Dan Dwyer's group, we did some measurements of the mast cells using flow cytometry. And you can see here that mast cells increased significantly in the lungs in response to house dust mite administration in both the control and the PGE insufficient strains, but it's about 2-fold more mast cells, real mast cell hyperplasia in the PGE synthase knockouts. Now, I said before that you can identify connective tissue mast cells and mucosal mast cells in the mouse, the latter being the induced population that arrives in the epithelium, sort of like the MCT population in humans. And you can identify these 2 subtypes based on 2 markers, beta-7 integrin, which is negative on the connective tissue mast cells and positive on the mucosal mast cells, and MMCP1, which is a gorgeous marker of the cells. Once they arrive in the epithelium, they turn on this protease gene, and you can readily detect them. So there's a big increase in this model in these inducible mucosal mast cells, and these account for the majority of the cells that crawl into the lung and the majority that overshoot in the PGE low situation. So that's all well and good. So what could we learn about IL-33? We started to wonder about factors that control IL-33, because IL-33 is a danger signal, and you don't want it sitting around in the tissues for very long. Otherwise, there'll be a lot of pathology. So it turns out that the IL-1 RL1 locus encodes 2 proteins. The cell surface receptor for IL-33 is called ST2L. And then there's a truncated protein called soluble ST2. These emerge from alternative transcripts. And soluble ST2 has the potential to bind up free IL-33 and prevent its binding from the cell surface receptor. There's surprisingly little written about soluble ST2 in the asthma literature, but Erin Gordon had a beautiful paper in JCI Insight a few years back where she studied the impact of 2 of the risk alleles in IL-1RL1, 2 SNPs, which correlate in human subjects with severe asthma and eosinophilia. And in these individuals bearing these SNPs, there is reduction in serum soluble ST2 and a diminished capacity for their epithelial cells to generate soluble ST2. So this implies that the control of soluble ST2 may be an important variable in terms of determining disease severity and maybe disease phenotype. So we decided to have a look at soluble ST2 levels in our mouse model. And at first we were shocked to see that while the control mice developed this nice increase in soluble ST2 in the BAL fluid in the lung tissue, we really didn't see that in the PGE synthase knockout. It was very, very blunted. And this led us to suspect that PGE might actually be directly inducing the production of soluble ST2. So we delivered PGE down the airways of these PGE synthase knockouts and showed that we could restore soluble ST2 by giving PGE down the airway, and it also decreased the indices of type 2 inflammation, as you might suspect. So we wondered whether the deficit in soluble ST2 was due to the deficit in PGE, because obviously this would have some implications for PGE low states in human disease potentially. So we delivered soluble ST2, recombinant protein, intraperitoneally to these PGE synthase knockouts and showed that indeed it reduced the indices of type 2 inflammation to levels comparable to what we had seen with the wild type control mice. So, does PGE control soluble ST2 production in humans? And the answer is probably yes. So we worked with our colleagues Tanya Laylaw and Katie Buchheit, who run a first-rate translational program focused on AERD, and they had samples left over from 2 of their studies in AERD subjects who had already had urinary prostaglandin E metabolite levels done. And we measured serum soluble ST2 in the serum samples from these subjects and found that there was a direct, positive, significant correlation. But more importantly, when we looked at soluble ST2 levels, we found that they correlated inversely with SNOT22, a measure of severity of sinus disease, with ACQ6, directly with percent predicted FEV1 and FVC, and inversely with blood eosinophils. So it appears that PGE may be regulating soluble ST2 as a mechanism that controls the strength of the inflammatory response in a way that relates directly to clinically important outcomes. So naturally, we wanted to know more about how this worked. Where is it coming from? And to make a long story short, we think it's coming from the mast cell. Remember, I said mast cells were the principal site of IL-1, RL-1 mRNA expression in both the airway and the sinonasal tissues. We didn't know from this level of resolution whether most of this transcript reflects the short versus the long isoform. And in collaboration with Dan Dwyer's group, we did some RNA sequencing on human cord blood mast cells that were stimulated for 72 hours with prostaglandin E. And we were very surprised to see that prostaglandin E up and down regulates thousands of transcripts. It's not just an inhibitor of activation, it's actually a reshaper of how the cell perceives its environment. And one of the things it does is it upregulates the expression of the short transcript of IL-1 RL-1 that encodes the soluble receptor while downregulating the long transcript. And to prove that this was actually functionally the case, we did some measurements of soluble ST2 production by mouse and human mast cells ex vivo using IL-33 in combination with PGE2, which gave us the biggest response. And you can see PGE2 substantially potentiates soluble ST2 secretion. This is a lot of soluble ST2, and it downregulates the surface receptor. So it changes the way the cell sees the IL-33. It also is markedly inhibitory for IL-13 production by the mast cells from both species. So are mast cells really the important source here? We used a pair of mast cell deficient strains of mice to answer that question. The Cree master mouse, or CPA3 Cree mouse, lacks mast cells altogether. And these mice have a hyperinflammatory phenotype when you give them dust mite, which is a little counterintuitive, except when you consider that they don't make much soluble ST2. It's markedly impaired, and that is reflected not only BAL fluid and lung, but also serum. We wanted to know which mast cell subsets were doing this, and conveniently, there's a mouse that lacks only the connective tissue type mast cells called the MMCP5 DTA Cree mouse. So these mast cells have no constitutive, no connective tissue mast cells, but they can recruit the beta-7 positive mast cells to the lung. And accordingly, these mice had no differences from the wild type in terms of eosinophilia or ILC2 expansion, and they had no differences in terms of soluble ST2 production. So the inference would be it's probably that inducible mucosal mast cell population that's making soluble ST2. And to further nail that down, we did some ex vivo experiments with sorted mast cells out of the mouse lung and showed that only the beta-7 high inducible mucosal mast cells could generate soluble ST2 in response to IL-33 and PGE. Now, is this true in the human? We're somewhat constrained by cell numbers, but using a novel in vitro system that Dan Dwyer's group developed, we were able to study MCT and MCTC human mast cells derived from circulating progenitors. And you can see very clearly it's the MCT mast cells that have this response, both to IL-33 itself and to PGE. There's an additive response when you put them together. And there's even sort of a constitutive leak in the absence of stimulation. I think what this says is that intraepithelial MCT population that is present in all type 2 high diseases may actually have an important role in mitigating the biggest problems that are associated with excessive IL-33 ST2 signaling. But in a PGE low background, that function may be taken away from the mast cell. And to further nail that down, we made a strain of mouse that lacks the ability to express either soluble ST2 or membrane-bound ST2 only in mast cells. And we studied these mice in both the PGE insufficient and PGE sufficient background. We got completely opposite results. If the PGE is low, taking away IL-33 receptor from mast cells protects against inflammation, ILC2 expansion, mast cell expansion, and lung IL-33. But if the PGE levels are sufficient, the opposite happens. It looks exactly like the PGE insufficient and the cremaster mice. The point being, PGE concentrations are super important determinants of whether the mast cells are a good guy or whether the mast cells are a bad guy, at least in these preclinical studies. Okay, so in the last few minutes, I'm now going to talk about some lessons we've learned from clinical trials. Omalizumab, which is not mast cell specific, has been around for a very, very long time. And this is from the work of Larry Schwartz and his colleagues who studied the genomic DNA of the patients who were enrolled in the original omalizumab phase 3 trials. It turns out that when we inherit the chromosomes from our mom and dad, we inherit 2 tryptase loci from dad, 2 from mom. Many of these tryptase loci bear mutations that render them non-functional. So some of us have only 1 or 2 active tryptase alleles in our mast cells. Some of us have 3 or 4, and that does correlate with tryptase levels in the serum. What these investigators showed is the entire efficacy signal in omalizumab is due to those individuals with 1 or 2 alleles. They have a significant bump in FEV1, which is not appreciated in the group as a whole. They have significant improvements in beta-2 agonist use, in asthma symptom scores, and asthma control. The folks who have 3 or 4 alleles absolutely have no response. So this indicates that the genotype of the mast cell may be an important predictor of how the clinical response to a mast cell-directed therapy might work or not work. And this is something that we have not taken into account in a clinical way. Now, another clinical trial was the KIA study that Elliott Israel really did in collaboration with our group and several groups across the country. This was a study looking at the impact of the mast cell depleting drug imatinib, which is a KIT tyrosine kinase inhibitor that also inhibits platelet-derived growth factors. So it's not entirely mast cell specific. The subjects all had refractory asthma without regard to whether they were eosinophilic or not. And the primary outcome was a change in doubling dose of PC20 to methacholine. And you can see that the imatinib group achieved a significant increase in PC20 by 3 months that was maintained out to 6 months. You see the placebo group drifting up a little bit probably because of adherence to their high-dose inhaled glucocorticoids and other treatments. There was also a statistically significant, although probably clinically not very relevant, change in FEV1. There was a reduction in serum tryptase, as you might expect, and a reduction in mast cell counts, including the airway smooth muscle compartment, where you remember you have those MCTC mast cells that correlated with airways hyperresponsiveness in Ian Pavord's studies. You do see a change in the placebo as well, which by the eyeball test isn't quite as impressive, but as a result, these 2 bars are not statistically different. And there's suggestion in the post hoc analysis that the imatinib may have been more efficacious for those individuals who did not have blood eosinophilia or who had higher levels of BAL fluid neutrophils than the rest. And this is a hypothesis that KIT would be better for type 2 low asthma that Elliott, I think, is pursuing in the PRECISE trial. There was another tyrosine kinase inhibitor subjected to a phase 3 study, the masitinib trial. This is a tyrosine kinase inhibitor that also inhibits KIT and platelet-derived growth factor. The primary outcome here was a reduction in annualized exacerbation rates across a range of asthma subtypes, you can see in general there was a pretty decent reduction, statistically significant, in the range of 33 to 38%. Unfortunately, in this study, the investigators found that a lot of patients did not tolerate this drug because of GI toxicity. That's not something that we ran into with the Kia study. Okay, so now I'm going to summarize and then leave some time at the end for Q&A if there are any. Mast cell hyperplasia and mast cell redistribution are features of asthma that correlate with specific disease surrogates. In the epithelium, you see hyperplasia in type 2 high disease. In airway smooth muscle, you see hyperplasia in both type 2 high and low. Mast cell activation surrogates correlate with severity regardless of endotype. This is not just an IgE and allergen story. There are multiple activation pathways brought to bear on the mast cell. I've talked about 2 of them today. There may be others that are just as important that we haven't yet discovered. Inhibition of KIT and platelet-derived growth factor depletes mast cells and generally improves airway function and disease control in severe asthma, but is not selective for mast cells per se. Not all mast cells are necessarily deleterious. There's the story of the small airway kinase positive mast cells that may be protective. And there's also this story about the prostaglandin E pathway for driving soluble ST2 by intraepithelial mast cells. I think it's likely that ST2 and IL-33 targeted drugs may be most efficacious for those individuals who fall into the PGE low or soluble ST2 low patient groups. I think this is potentially an important biomarker that might be brought to bear in clinical trials. And I think the extent of mast cell functional heterogeneity in the airway in general and asthma in particular is still to be determined as the tools move forward and we get better and better at working with smaller and smaller cell numbers. So, to thank the people who really did the work involved. Keenan Al-Halak is the bearded gentleman in the front row of my lab group here, and all my lab group contributed to his study. I have the good fortune of a long-term collaboration with Elliot Israel and some really wonderful collaborators within my own division, Katie Bookheit, Tanya Laidlaw, Patrick Brennan, Dan Dwyer, and Nora Barrett. And of course, I want to thank the NIH for funding these studies. May it live on in perpetuity. And I'm happy to ask or answer questions. Thank you for your attention.
Thank you, Josh. That was a really wonderful talk. It was just so sweeping in scope, starting from Ehrlich and going to the latest techniques in terms of cell sorting and expression. And thank you for that really major overview and the really wonderful work that you and the collaborators that you're working with are doing. So there's a question in the chat. So if you could give PGE2 to asthmatics, either by inhalation or intravenously, or both, would you consider that? And why hasn't that been really pursued?
That's a beautiful question. Well, I wouldn't give it intravenously because prostaglandin E receptors are all over the body, including the vasculature and other places. And you might get into some serious problems with blood pressure control, heart rate, etc. So I think you would want to deliver it locally. The issue with delivering it locally is twofold. One is that prostaglandin E is very unstable. So you would require some sort of very frequent dosing to get efficacy, I think. The other problem is it's irritating. It makes people cough. And all of these inhalation studies that were done back in the early to late 90s were restricted by coughing. We have good evidence from the guinea pig, whose airway is hooked up a lot more similarly to human than the mouse airway, because mice don't cough, that the EP3 receptor is responsible for the cough reflex, and that is neurologically mediated. If you could get around using an agonist that only stimulates EP2 or EP2 plus EP4, you might have some therapeutic advantages. Now, Ono Pharmaceutical did develop some very nice selective agonists, which we use in our preclinical studies. They have not been anxious, surprisingly, to put these into clinical trials in asthma, and I'm not sure what the basis of that reluctance is. June Nagai in my laboratory, who's a card-carrying molecular pharmacologist, is actually working on developing some biased agonists of the EP receptor system. So hopefully something will come out of those efforts.
Josh, another question. So you talked about ST2 and IL-33, and as you know, there was a clinical study with an IL-33 inhibitor looking at the effects in asthma, and it actually was effective, right? But it wasn't any more effective or additive as compared to dupilumab, which blocks IL-4 and IL-13. And considering the scope of what you talked about, one would have thought that it would have been. What do you think about why there didn't seem to be any increased effectiveness or additive effect when you combine the two of them together?
Sure. Yeah, no, I thought that was a very interesting study since both antibodies were made by the same company. And that was the main reason why they didn't move forward with their IL-33 program. They were hoping to get a synergistic effect. The only thing that the two things together did differently than either one alone was that the IL-33 eliminated the increased eosinophilia that you saw with the dupilumab. Which I think proves that IL-33 works upstream and controls probably the IL-5 axis. I think they're both incredibly important. It's just that one is upstream, very proximal to the immune response, and the other is downstream. Every important cell type has IL-4 receptors on them. I mean, all of them. So I think that's the reason for the sort of broad-based efficacy. I would love to see a study that takes into account the PGE biomarker, because my bet is that if you focused on that group, you might actually see some preferential efficacy from IL-33. That said, IL-4 receptor alpha signaling is suppressive of PGE production. And Katie Buchheit and Tanya had a beautiful Jackie paper a couple of years ago where they looked at biomarkers in the nasal fluid and the urine in patients with AERD who were in an open label study of dupilumab. PGE shoots up like a rocket after 1 month of dupilumab treatment. Now, those are patients, AERD is sort of a special case. Those are patients where there's a severe depletion of PGE. Maybe, dupilumab works really well for them. Maybe the anti-IL-33 would work really well too. But in terms of an advantage, I can't give you one at the moment.
In the case of where the PGE, you said the PGE in the nasal polyp situation really went up using an IL-4, IL-13 inhibitor. Why do you think that is?
So if you stimulate epithelial cells with IL-4 or IL-13, you downregulate expressions of cyclooxygenase 2 and prostaglandin E synthase. And that was from a paper by Sally Wenzel about 20 years ago. And that's been reproduced in other cell types, including fibroblasts. And I think that may be one of the ways that 4 and 13 have to sort of unbreak the system and permit some type 2 inflammation when it is necessary and useful to the host, but it never shuts down.
Another question is, can we measure PG levels in humans in the clinic, in the urine? Is that something that would be useful?
Well, there are ELISAs you can use to measure PGE itself. They're reasonable. They're not used commercially. They're for research use only. The urinary metabolites are the most accessible way to measure, but that requires mass spectrometry. So right now, there's no practical way to do that in the clinic. You could certainly do it in the context of a clinical trial, though.
Okay, we've reached the top of the hour. This was, again, just a wonderful, beautiful talk, Josh. If you'd like CME credit for this, please email eisraelasst@bwh.harvard.edu or eisraelasst@partners.org, and we can make sure that you get the CME credit. Thanks so much, and everybody have a wonderful day.
Okay, thanks, everyone.